Preliminary examination of genetics. Number 1. Define genetics and its importance in fisheries. It is very important in fisheries in terms of breeding program. Fish and breeding program will be crucial to this development, not only to reach the production goals but also to reduce production costs, improve disease resistance, improve utilization of feed resources, in improving product quality. It is the history of animal breeding. Sure, the principle of selection have been applied for thousands of years, long before the theoretical background was developed in considerable success. Number 2. What is the chromosomal and chemical basis of heredity? Chromosomal is the structure found inside the nucleus of a cell. A chromosome is made up of protein and DNA organized into genes. Each cell normally contains 23 pairs of chromosomes. DNA is the known of chemical basis of heredity. DNA is made up of a combination of nitrogenous bases, sugar molecules, and phosphate groups, which is linked by the different bonds in a series of sequences. Number 3. Explain the concept of population genetics. Population genetics is a branch of genetics that conduct the study of the genetic composition or composition of population. It combines genetics, evaluation, natural selection, selective breeding, statistics, and mathematics. It makes computer and mathematical models and conducts field research to test the methods. Population genetics spend their time doing two things: describing the genetic structure of population or theorizing about the evolutionary process that affect population. Number 4. What is the importance of selection in fish tank management? The undertreatment and the parameter estimate can be incorporated in the tank assessment to produce the variable solid under the given management scheme. This variability produces facility and manager, but also has implications for the stock depending upon where in the range of possibilities the manager depends. Number 5. Differentiate hybrid from inbreeds and plain or a good crossbreeding program. Hybridization is the process of crossing genetically different individuals to produce offspring, where inbreeding in the cross of two closest related parents who share very similar alleles. Inbreeding is more likely to cause genetic abnormalities unlike hybridization. Number 6. Identify biotical in fish genetics and determine how they could help increase productivity. biotechnology allows scientists to identify and combine traits in fish and shellfish to increase productivity and improve quality. Scientists are investigating genes that will increase production and natural fish growth function as well as the natural defense combined marine organism used to fight microbial infections. Number 7. Differentiate mistake from nuisance. Mitosis is a process of nuclear division in eukaryotic cell that occurs when a parent cell divides to produce two identical treated cells. Mitosis is the considerately divided into 5 stages known to proper apoptosis, metaphase, anaphase, and telophase. Mitosis is a process where a single cell divides between to produce 4 cells containing self-organizational amount of genetic information. These cells are our sex cell, sperm in the male, eggs in female during meiosis. One cell divided twins or two form of 4 daughter cells. Number 8. Differentiate DNA from RNA. DNA is a double-strand molecule, while RNA is a single-strand molecule. DNA is stable under alkaline condition, while RNA is not stable. RNA and DNA based on pairing is slightly different since DNA use 4 bases adenine, thymine, and cytosine, and guanine. RNA use adenine, uracil, cytosine, and glutamine. Number 9. Discuss how and why DNA replication occurs in their major step, the opening of the double helix and separation of the RNA. DNA strands and priming of the template strand and the assembly of the new DNA segment. During separation and two strands of the DNA double helix uncoil at a specific location, cell and organization. The RNA applicant because wherever a cell divides, the two new daughter cells must contain the same genetic information or DNA as a parent cell. Explain the importance of transcription and translation. The goals of transcription is to make a RNA copy of a gene's DNA requires for a protein coding genes. The RNA copy or transcript carry to information needed to blood of polypeptide proteins or proteins submit. Eukaryotic transcript need to go through same processing step before translation into proteins. Translation is enable to effective combination between people around the world. It is a career for the transmission of knowledge, a protector, and cultural heritage, and essential to the development of a global economy. Highly skills translation are key. Number 11. Discuss how proteins, amino acids, and cytosides. During translation, ribosomal subunits assemble together like a sandwich on the strand, where they process to attract the RNA molecules to amino acid. A long chain of amino acid emerges as, as the ribosome decodes to our mRNA requires into a peptide or a new protein. Explain the principle of YY male technology and the process on how to produce YY males. YY males produced by converting into male populations. Number 13. Explain the different kinds of artificial selection method, mass selection, family selection, and multi-trait selection. Mass selections. This kind of artificial selection, which is the simplest form, is also known as individual selections. In mass selection, the best individuals are selected from a population for breeding, and the remaining individuals are discarded in progeny of the selection individuals are usually better on average than the original population. Family selection. Family selection is to use when the heritability is small and or there the uncontrollable source of environmental viruses. It involves in chooses entire family, usually group of poor birds or survivors. Family Families are selected on the basis of their means populating value. Multiple trait selection. The best approach for selecting animal considering multiple traits is used on economic selection in box. The overall breeding goals each is a improve multiple combinically relevant traits by selecting animals using a selection in box. Number 14. Advantage of crossbreeding. Crossbreeding is used to produce superior fish for grow out production fish. Wool selection is used to create superior blood fish. One of the most common breeding programs in aquaculture is the production of sex ribbons proved stock to produce monosex population for grow out.